Hello there. My name is Brian Pfleger, and I'm a professor in the Department of Chemical and Biological Engineering at UW-Madison. This short video is meant to be an introduction to my research group. If you'd like to contact me, you can see my email address below. And if you want more information, you can visit our lab website at the address you can see on the screen. So first, a little bit about me. I grew up in southern New Jersey, just across the river from Philadelphia. There, I developed a strong love for sports, including ice hockey that I learned to play in high school. And it also gave me a love of all of the professional Philadelphia sports teams, including the Eagles and Phillies, who had fantastic years this year. So after high school, I decided to go to Cornell University to pursue a degree in chemical engineering. And upon the advice of my faculty there, went out west to sample the different lifestyle at the University of California, Berkeley. After finishing my PhD, I moved to the University of Michigan, where I was a postdoctoral fellow in the School of Pharmacy, studying natural products biosynthesis for 2 years. During my Cornell days, I had a research experience with 3M in St. Paul, and this was in the traffic control materials division, where I studied how diamond grade sheeting is assembled as a bilayer material. Now, this experience taught me two things. One, that I absolutely loved the research process and figuring out how to solve problems. And two, that I wanted nothing to do with materials research. So when I went to the University of California, Berkeley, I discovered the world of metabolic engineering, where we use the principles of chemical engineering to alter kinetics inside cells to adjust how they take resources in and convert them to desired products. Outside the lab, I do a little bit of my own fermentations in my own brewery in the basement of my house. I still play ice hockey, and I love spending time with my family, whether it's like this summer here in Germany, or hiking the Ice Age Trail in and around Madison, or taking my family to Badger football games like we did earlier this fall. And what do I like the most about UW-Madison? Honestly, it's the collaborative culture, the spirit that we're all working together to make life better for everyone else, the true Wisconsin idea. The balance of work and life, because Madison is this fantastic small city, a place where we can do great work, but also have a very high quality of life. And honestly, if you're looking for a place to study biotechnology like metabolic engineering, there are very few places better than UW-Madison. So a little bit about my research group. We primarily are made up of PhDs, about 2/3 PhD students and postdocs historically. We have 12 researchers currently. We're a fairly senior heavy group with a large number of students in their fourth year. Primarily, the PhD students come from the engineering disciplines, primarily chemical engineering, and we balance that with postdocs that primarily come from the natural sciences, microbiology and chemistry, or biochemistry being the most common. And you'll see that we have students from across the country and across the world, and that we're a pretty fun-loving group, whether it's out for Halloween, visiting a beer garden, presenting scientific posters at conferences, or just relaxing in and around Madison out on the waterways. So our research can be described in many different areas. And so what I did was I created this word cloud that sampled all of the different things that we've done in or published in our papers. And as you can see, the big words tend to be E. coli, fatty acid, gene, cyanobacteria, and gene expression here being the key component, as well as metabolic engineering. And so you can see that those are the threads that go throughout our research. So what are these particular fields? Well, in general, what we're looking at trying to do is create sustainable chemical processes. So that means instead of having a linear flow of carbon from, say, fossil resources to our desired final products, we have something where we take carbon dioxide and use solar energy to produce those reduced carbon molecules that we can combust and take some energy out and replace the CO2 that we need to close this in a cyclic process. Now, this can be done many different ways, right? With traditional heterogeneous or homogeneous catalysis, with electric catalysis. But my interest is in biocatalysis. And so what we're going to do there is combine the sets of reactions catalyzed by enzymes inside cells for the most part. And so what this means is we take the metabolism as we represent here in this set of reactions going from A to C, and we augment that with, say, new reactions that we're interested in, or we eliminate other reactions that we don't want to have happen based on the natural repertoire inside an organism. And so this type of work requires the development of different types of tools. So we need to understand which enzymes are going to be the best, If we can discover those, we'll use them. If we can evolve them, we'll do so. And in other cases, we have to engineer them directly to improve their properties. In other cases, we'll need to then take those enzymes and put them in combinations with regulatory systems to control when the proteins are produced in the right conditions. And then once we built these first generation strains, we perform fermentations to see how well they've done, and then use the tools of systems biology to understand where we're currently bottlenecked. And so we'll use things like functional genomics, flux balance analysis, and other types of traditional kinetic models. And so while we've started our work primarily in E. coli, we've developed a lot of tools for cyanobacteria like Synechococcus and Pseudomonas putida. We're now moving into other organisms, including yeasts, as well as other bacteria that have very useful or unique properties. This is the power of the modern synthetic biology era, where we can now do the things we used to do only in model organisms with virtually any organism of our interest. And so our work is currently funded primarily from the Department of Energy. And so we have projects directly to look at the metabolism of cyanobacteria, the production of oleochemicals in oleaginous organisms like Euroia lipolytica, as well as the production of biofuels through the Great Lakes Bioenergy Research Center. We're also producing chemicals in a CO2 neutral way in collaboration with a company called Lonzatec in Chicago. Lonzatec's expertise is in the production of acetate and other small organic molecules from gas fermentations, meaning taking CO2 and hydrogen and producing those small molecules. And then we're upgrading those intermediates into useful reduced chemical products. And in addition to synthesis, we are studying other aspects of the environment and how we can use synthetic biology to improve things like phosphorus wastes in the environment from manure. This is a collaboration actively with Professor Zavala in the department and other professors around the Madison campus, as well as engineering microorganisms in the soil to try to enhance the ability for agriculture to produce higher yields with less chemical fertilizers. And so this is in collaboration with Professor Venturelli and other experts in the rhizosphere microbiomes. So one recent story I wanted to highlight is how we can combine gene discovery with engineering principles to apply for producing chemicals in a sustainable way. And so this was a process that started with the idea that this molecule, libulinic acid, which is a 5-carbon keto acid, this is a molecule that Professor Domesic in the department had shown that they could make from different types of biomass quite reliably. And it's a platform chemical that we thought we might be able to upgrade into other types of useful chemical products. The challenge in the biocatalysis world, though, was we did not have a biosynthetic pathway or a catabolic pathway, one that could consume the libulinic acid into useful building blocks. And so Jackie Rand, a recent graduate of the lab, spent her PhD doing a combination of genetic, in vitro reconstitution, and metabolite reaction analysis to isolate the genes in Pseudomonas putida that were capable of doing this catabolism in that particular cell. And so she was able to identify an operon of genes inside the genome and assign the biochemical function to each of those reactions. And so they fit this nice yellow pathway where we activate our organic acid as a coenzyme A thioester. We reduce it, the ketone at the 4 position to a hydroxy. We activate it, which is the unique step here with a phosphorylation event that gets dephosphorylated to isomerize the hydroxy from the 4 to the 3 position. And now this 5-carbon 3-hydroxy-CoA is an intermediate in beta oxidation. And so this molecule can then be split into acetyl-CoA and propionyl-CoA, which are central cellular building blocks. Now, you would think that if we just put this set of genes inside E. coli, we would now have the ability for E. coli to consume libulinic acid. And it turns out that that wasn't true. We actually had to adaptively evolve a strain in order to allow it to use this artificial or heterologous pathway. And along the way, we discovered that it was producing small quantities of this 2-butanone instead of the acetyl-CoA and propionyl-CoA that we were desiring to make. And so Chris Mayer realized that perhaps this was an opportunity to, instead of producing the central building blocks, produce 2-butanone, which is also known as 2-methyl or methyl ethyl ketone. It's a common important industrial solvent. And so what he decided to do was take a look using systems biology models at how this particular bioconversion could be performed. And he discovered that the way that the butanone was coming was when acetate was present in the media, was converted to acetyl-CoA by transferring the coenzyme A off of this 3-keto-CoA, ketovaleryl-CoA species. The resulting 3-keto-valerate would spontaneously decarboxylate or could be accelerated with an additional enzyme to produce the methylethyl ketone. And so when he looked at the metabolism in genome scale metabolic models, he saw that the levulinate and the acetate could basically create a large space where either you could make biomass, make these products, or you could make all butanone with no biomass being generated. And so what this ends up with then is a bioconversion where the acetate, even if it's fed in excess, is consumed first, and it doesn't allow the cell to consume all of the levulinate that's needed for this process. If, however, we delete the genes that allow for acetyl-CoA production, now the only way that acetate can be converted to acetyl-CoA is through this particular reaction that also produces our 2-butanone. And so when you model that, you see this new space. And so for cells to produce any biomass or any energy, they have to produce the butanone at its maximum rates. And so now you'll see that in the excess acetate case, we still consume all the levulinate first, and we don't touch the residual acetate. And so this allowed us to create a process where we could get unit conversion of the levulinic acid from biomass sources directly to butanone in 100% yield. And so for most of our work, as I mentioned, is funded by the US government. We do have some projects that are funded with collaborations with companies. That's built some strong relationships, primarily through my former students who have gone on to work at just about every one of the major synthetic biology companies in the US. And we have had a few folks who have gone on to academic careers as well as careers at the various national labs, both in the US and abroad. And so if you're interested in learning more about my group, please feel free to reach out and contact me or my students at either of these 2 locations. I look forward to meeting you in the process here and hope you decide to join us here in Wisconsin. Cheers.